Welcome to this explainer. Today, we're stepping into the shoes of a very specific, kind of fascinating type of detective. We aren't hunting for clues at a typical city crime scene, you know? We're actually hunting for totally invisible culprits in the microbiology lab, a place where a single tiny glass slide can literally hold more suspects than the population of a small city. Every single day, medical microbiologists act as microscopic detectives, trying to unmask organisms that are completely invisible to the naked eye. To pull this off, they rely heavily on bright field microscopy, which basically means illuminating specimens with visible light to form images based on differences in light absorption. But here's the thing. Just looking through a lens isn't enough. We have to actively force these microscopic suspects to reveal their true identities, their weaknesses, and their hidden weapons. So, here's our roadmap, our Microbial Detectives Toolkit. We're going to cover 1. Securing the evidence, 2. The primary interrogation, 3. The tough suspects, 4. Hidden weapons and structures, and finally, 5. The raw evidence. Let's get right into it. Section 1. Securing the evidence, microscopy, morphology, and smears. Before we can start interrogating anybody, we need to bring them into the light and establish exactly what we're looking at.
Okay, so the absolute crucial starting point is knowing the difference between the physical shape of a single bug, which we call its morphology, versus how the whole gang hangs out together, which is its arrangement. Think of morphology as the suspect's basic physical description. It tells us the structural appearance of the individual organism. Arrangement, on the other hand, is kind of like their gang sign. It tells us exactly how those cells are positioned relative to one another.
Think of these as the classic mugshots of microbiology, the basic shapes and groupings we use to start profiling. For shapes, we've got the spherical cocci, the rod-shaped bacilli, the comma-shaped vibrios, and the rigid spiral spirilla. But they rarely work alone, right? Their arrangement is just as telling. Cocci, for example, might form pairs, which we call diplococci. They might form long chains, known as streptococci, or even these distinct grape-like clusters known as staphylococci. Getting this initial profile right is absolutely step one of the whole investigation. Now, to actually secure this evidence so we can study it, we have to follow a really strict set of preparation steps. You take your specimen, place it on a clean, labeled glass slide, and this part is vital. You prepare a thin, even smear. Why thin? Well, a thin smear allows our chemical stains to penetrate properly later on, and it makes it so much easier to clearly read that cellular morphology we just talked about. Once it air dries, we fix it using heat or chemicals. This fixation step preserves the morphology and literally glues our suspects to the glass so they don't just wash away during the chemical interrogation. Moving on to Section 2, the primary interrogation, the critical Gram stain. This is where things get really interesting because this differential staining technique divides bacteria into two massive camps based entirely on their cell wall structure. If you want to think like a microbiologist, you need to lock this memory trick into your brain. It's an absolute cheat code. CIDS. Crystal violet, iodine, decolorizer, and safranin. This is the exact order of the reagents used in the Gram stain, and it's the foundation of almost everything we do. Imagine this procedure unfolding step by step, taking our invisible cells through a carefully designed chemical gauntlet. Step 1, we completely flood the specimen with crystal violet, our primary stain, which turns all the bacterial cells a deep purple. Step 2, we add Gram's iodine. This acts as a mordant, forming a tight chemical complex with the crystal violet deep inside the cell. Step 3 is the decolorizer, usually an alcohol or acetone mix. And finally, Step 4 is the safranin counterstain. So what does this actually do? It's brilliant chemical sorting in action, and it is completely dependent on the suspect's armor. Gram-positive bacteria have a super thick peptidoglycan cell wall. It's like heavy molecular chainmail. During that decolorization step, this thick armor retains the crystal violet complex, keeping them a vibrant, unmistakable purple. But Gram-negative bacteria, they have a much thinner layer of chainmail and an extra outer membrane. The decolorizer strips the primary stain right out of them, leaving them totally colorless, until they take up that pink or red safranin counterstain. Purple equals positive. Pink equals negative. This is the foundational language of the clinical microbiology lab. If a doctor is desperately waiting on a crucial lab result to figure out which antibiotics to prescribe to a very sick patient, this is literally the very first thing they want to hear.
So let's test your detective skills for a second with a classic exam question that actually has huge real-world patient consequences. Out of the 4 steps we just walked through, what would you say is the most critical step in gram staining?
If you guessed the decolorization step, you hit the nail on the head. Because slipping up right here gives your suspect a completely false identity, which leads to catastrophic lab errors. Think about it. If you over-decolorize, your thick-walled, gram-positive organisms will lose their purple dye and appear falsely gram-negative. But if you under-decolorize, your gram-negatives hold onto the purple and look falsely gram-positive. Add in real-world variables, like accidentally making your initial smear too thick or using a really old bacterial culture, and you get totally unreliable reactions. Precision here is absolutely everything. Section 3. The tough suspects. Acid-fast bacteria and the Ziehl-Neelsen stain. You know, sometimes our standard interrogation tactics completely bounce off, which means we have to call in a specialized approach. Some organisms totally ignore the gram stain. They have heavily armored cell walls packed full of mycolic acids, acting almost like a thick, waxy raincoat. Normal stains just beat up and roll off. So we have to literally steam the primary stain, carbonyl fusion, right into them using gentle heat, being careful not to boil or dry out the specimen. Once that intense red stain is finally forced inside, their mycolic acid armor actually turns into a defense mechanism for us. They will fiercely resist the incredibly harsh acid-alcohol decolorizer we hit them with next. Finally, we wash and counterstain the rest of the sample with methylene blue. The visual payout for this specialized tactic is just striking. The acid-fast bacilli, or AFB as they're called, end up glowing like bright red beacons against a cool blue background. Anything that is not acid-fast takes on that blue counterstain, making the incredibly dangerous red bacilli, like Mycobacterium tuberculosis, really easy to spot. Now, as a cool, modern upgrade to this classic method, labs can also use fluorescent stains, like auramine-based fluorochrome stains. When viewed under a fluorescence microscope, it's basically the microbiological equivalent of flipping on night vision goggles. It takes that red beacon concept and literally makes the organisms glow brilliantly in the dark, which massively speeds up the hunt for these dangerous pathogens when you're scanning a slide. Section 4. Hidden weapons and structures, spores, capsules, and the Albert stain. Bacteria aren't just simple geometric shapes. They often carry biological armor and survival kits that we can only see if we use highly specific staining tricks. Take the Albert stain, for example. We use this to identify specific, dangerous targets like Corynebacterium diphtheria, the bug responsible for diphtheria. This stain specifically lights up its metachromatic granules. You can think of them almost like unique internal fingerprints. Under the microscope, the main bacterial body takes on a green color, while those telltale metachromatic granules pop out in dark blue or black. It's an incredibly targeted profiling tool. Then there's the fascinating reverse psychology approach of negative staining. Sometimes trying to stain the organism itself just doesn't work, especially if it has a capsule. A capsule is this slippery, sugar-rich external layer that perfectly protects the bacteria from our immune system. So instead of fighting it, we use India ink to paint the background completely black. The organism itself remains unstained, which reveals the clear, glowing halo of its dangerous capsule set dramatically against the dark background. And for the ultimate microscopic survivalists, genera like Bacillus and Clostridium, we have the Schaffer-Fulton spore stain. These bugs produce highly resistant structures called endospores to survive extreme conditions. To unmask them, we use a primary stain of malachite green, which locks tightly into those tough endospores. We then counterstain the rest with safranin. The end result? The highly resistant endospores appear a vivid, striking green, while the normal, vulnerable vegetative cells take on a soft red or pink color. Finally, we reach Section 5, the raw evidence, KOH, and wet preps. Sometimes the absolute best way to catch a suspect isn't with complicated chemical stains, but by observing them in their raw, unfiltered environment. We're talking about direct windows into the wild microscopic world. Whether we are literally melting away human skin scrapings, hair, or nail clippings with a harsh 20% potassium hydroxide, or KOH prep, which I know sounds totally brutal, but it perfectly digests human keratin to clearly reveal intact fungal hyphae or yeast cells. Or, just using a simple saline wet mount to catch motile organisms darting around live in action. We can even use iodine wet mounts to enhance the visibility of internal structures in stool specimens for parasitology. It's all about catching the organisms in their most natural state. It's truly an incredible process, isn't it? From the basic bright field microscope to the unforgiving chemical gauntlet of the Gram stain, right through to the highly specialized techniques meant to break down waxy armor. Thank you so much for joining me on this explainer today. I'll leave you with this final thought. With so many protective layers, tough cell walls, and hidden survival structures already uncovered by these dedicated detectives, what other microscopic secrets do you think are out there, just waiting for the right stain to be revealed?